Hi, today we're going to explain the theory of DNA plasmid isolation from bacteria. All isolation procedures require identification of unique characteristics in the target. For example, if wanting to isolate a triangle and given a square, triangle, and circle, we could first isolate for shapes containing corners and then select for shapes containing a certain number of corners. All cells contain four major macromolecules: nucleic acids, such as DNA and RNA, carbohydrates, lipids, and proteins. We want to isolate DNA, so we will focus on nucleic acids. DNA has many identifying characteristics, allowing for successful isolation. Specifically, DNA is double-stranded, being held together by hydrogen bonds. This property allows for the separation of strands using high pH or high salt concentration. DNA also has a polar negatively charged backbone, which allows for solubility in water. DNA is also very large, which increases the likelihood of aggregation and promotes precipitation upon disruption of the dielectric constant, for example, when adding ethanol. Lastly, plasmids, which are our focus, are much smaller than genomic DNA, which allows us to separate based on size. There are three basic steps in plasmid isolation: cell lysis, plasmid isolation, which is most of the work, and finally, plasmid collection. In order to lyse the cell, a strong base is added in conjunction with a detergent, in this case SDS. This will disrupt the lipid bilayer, expelling the contents of the cell and denaturing both the plasmid and genomic DNA. Isolation begins with the removal of genomic DNA and other contaminants, such as protein. An acid is used to rapidly drop the pH. Small DNA molecules, such as plasmid DNA, will reanneal and enter back into the solution, whereas large chromosomal DNA fragments will not successfully reanneal and form a precipitate. The precipitated material can be removed via centrifugation. At this point, large impurities, such as proteins, are removed. Following centrifugation, the supernatant is added to specifically designated silica-coated columns, which function to retain the plasmid DNA through undescribed interactions. The silica column with bound plasmid DNA is washed with 70% ethanol, which removes other small charged contaminants which are bound to the silica membrane. Due to DNA having lesser solubility than these contaminants, the plasmid DNA is not removed from the membrane during this wash step, while other, more soluble contaminants are. Pure water is used to solubilize plasmid and thus remove it from the silica column. To perform a plasmid isolation protocol, you will need a stock culture bacteria, a spin column, an extra tube, a centrifuge, and a set of reagents from a plasmid isolation kit. First, centrifuge your stock bacterial solution at max speed for a few minutes to create a pellet. Next, remove the remaining supernatant from the pellet. Add resuspension buffer to your bacterial pellet and resuspend by pipetting up and down several times until a homogeneous solution forms. Once homogeneous, add lysis buffer, mix gently, and incubate at room temperature for 5 minutes. After letting the solution incubate for 5 minutes, add precipitation buffer and mix immediately by flicking the tube or inverting it up and down. Do not vortex at this step. Immediately after mixing, centrifuge the lysate at maximum speed for 10 minutes. Remove the resulting supernatant containing your plasmid and put it into the spin column. Be careful not to disrupt the pellet. Once the supernatant has been added to the spin column, centrifuge at 12,000 Gs for 1 minute. Discard the resulting flow-through and add wash buffer onto your spin column. Repeat the previous step in which the spin column is spun for 1 minute at 12,000 G and the flow-through is discarded. Add a new recovery tube to the bottom of the spin column. Add TE buffer to your spin column. Let the TE buffer incubate with the spin column for 1 minute. Finally, centrifuge the spin column with the collection tube at 12,000 Gs for 2 minutes. The resulting solution in the collection tube contains purified plasmid DNA.